So what we're looking at here is immobilizing the drug of interest, in this case a monoclonal antibody, onto the chip surface, then injecting serum samples from patients to look for serum antibodies in those patients. To the left, your left here, the red scale here is for the ELISA, the blue one here is for the BioAccord. So the blue bars is following the immune response progression in those patients, and the red ones here are for the ELISA. And what was quite interesting here is that really after only 2 weeks you can start to see the presence of anti-drug antibodies using the label-free method, but only after 12 weeks you can start to detect antibodies using the ELISA-based technology. And some of you may then say, why and how is that the case? The ELISA is usually quite sensitive. Well, usually for this type of persistent response or for a transient response, in the beginning of a treatment you have low affinity antibodies, and if you're having a label-based approach with, for example, an ELISA, you need to do wash steps, incubations, and you miss those low affinity interactions usually. And that can be quite significant because some of those early responses can have a clinical effect, and that has been shown by Amgen. So it has been actually recognized to the effect that Amgen today are labeling the drugs BioAccord tested. So you can find BioAccord results on the label of the Vectibix drug product on the market, for example, because they find that this is an additional testing requirement for their drugs they're putting to the market to show that they have monitored also the low affinity responses. Of course, if you were looking 10 years back, many clinicians would say that low affinity immune responses does not matter, and often that is the case, but not always. And as the FDA says, safety first. You need to find those antibodies, and you need to monitor the progression of that immune response. Also, with the BioAccord technology, you can quite rapidly characterize the type of immune response, and that is of course important when you're monitoring your individual patients for biotherapeutics, but it is of paramount importance when you're looking at efficacy developing vaccine products. In this case here, we are looking at an immunotherapeutic from an Uppsala-based company called Resistencia Pharmaceuticals. What they're doing here is putting down a receptor on the surface. In this case, it's the Ig receptor. This immunotherapeutic here was used to treat asthma and allergy in patients. So they are basically injecting this immunotherapeutic in the patient population, monitoring immune responses that can neutralize circulating IgE. In this case here, they're doing a preclinical study with rats, and they inject the serum sample from the rats and they monitor the immune response. The increase in binding level here is just showing a detection of a polyclonal response. Then by injecting isotyping reagents, you can quite rapidly characterize that response. And as depicted by this binding curve sensorgram, you can see that when you inject the anti-IgG2A, you have another shift of baseline, indicating that predominantly this immune response is an IgG2A response. And this is very important for the formulation and the evaluation of adjuvants for vaccines. And additionally, with the biosensor, you can also look at the neutralization capability from a binding perspective for certain type of vaccines. As shown here, you can look at the dissociation of an immune response directed to a pathogen or virus, and you can look at slow dissociation from certain preparations of a vaccine. So this is a very important qualitative information for your design and formulation of a vaccine. And on the contrary, looking at the biotherapeutic, you don't want to see that kind of progression of your immune response, because that may indicate a more robust immune response for a biotherapeutic on the other hand. So you can be using them for variety of purposes, looking at biologics. And I think this is one of our most and fast growing areas for the use of SBR biosensors today. Okay, so moving forward in the drug development process, looking at biotherapeutics, I just would like to show you the capability to measure with high reproducibility in other complex matrices. And this is an example from a biotech company called Micromet AG in Munich. They are, as some of you may be aware of, developing antibody-based therapeutics. In this case here, we are looking at the MT201. The MT201 was a candidate against prostate cancer. And And this is a process step, This is a process step where you are looking at cell culture supernatants, and this is a purification step after the antibody isolation. It's a mAb select sure step in the purification process. And the brownish bar here represents the BioCore data, and the red one here, the ELISA data. And as you can see, the reproducibility and the precision of the biosensor is sometimes an order of magnitude better than the ELISA-based assay. And that is important from a control perspective when you're looking at optimizing either a target purification to optimize the yield of a target, or for biotherapeutic purposes where you're trying to monitor your product and do product characterization over time. Another application is, of course, comparability, biocomparability, to use the binding information to look at post-translation modifications in a process, or when scaling up from a low nanogram scale to a milligram scale during development of proteins, whether that is targets or biotherapeutics, doesn't really matter. Here I would like to switch gears a little bit and talk about the use and the complementary use of microcal technology, and in particular DSC, differential scanning calorimetry. As you know, DSC is actually measuring the heats of interaction. And in this case here, we are looking at the VP DSC system. It's a fully automated system where you're monitoring the conformational change of a protein when you're heating up your protein in a cell over time. And we're looking at the melting points, the TM, the transitional midpoint. So basically, when you're heating up your protein under a certain condition, you can see the protein unfold, and at a certain point you have 50% folded and 50% unfolded, and that is the TM midpoint. So whether you are imposing intrinsic changes to a protein, you're changing something to your structure, or whether you are doing extrinsic modifications, you're changing the buffers around your protein, you can confer stability to a protein, or you can defer stability depending on the situation. But in either case, the TM is an indicator of stability of that protein. So this is basically how it works. You're heating up your protein, you're monitoring the heat capacity over temperature. So if at one buffer condition you have that TM around 70 here, if you change the buffer composition, you can see a shift in the TM indicating a more stable protein. This is a very straightforward method, and I will not go into the technology into more detail than that. Instead, show you one example here. This is from a pharma company where they're using the DSC to look at different tagged proteins in the purification process. It's actually a target protein. They're looking at selecting the protein that they can use for crystal growth in their structural studies, and quite quickly they can look at different variants here with different his tags, and if you're monitoring the TM for those different tagged proteins, you can quite quickly find that the untagged protein here has the highest TM, and that also later showed to be the best soluble protein, the most stable protein, and the most suitable one for crystal growth. So this is one application. The other one is when you're purifying a target protein or a biotherapeutic, and I would like to show you one example here from a US-based company called Diosynth, now part of Schering-Plough. In this case here, they were actually developing a process for a client. Diosynth is a CMO. They were developing a purification process for a monoclonal antibody. The issue here was that their protein A resin here had a pretty low capacity, and the protein was starting to aggregate in the elution buffer. That meant that they really couldn't get any efficiency in this separation. And to make things worse, you elute your column at a low pH, which further caused precipitation and aggregation of the protein in this process. To that extent that they needed to have a filtration step at the end to concentrate the protein. And as you can understand, when you're scaling up, this adds a lot of capital cost to your process. So they decided to try to see how they could stabilize the protein of interest in the elution buffer. And to do that, they utilized the capability of DSC. So they started with a buffer of citrate pH 3.5, changed conditions, and found after a few days that the additional mannitol at the same pH actually shifted the TM to the right, to a higher temperature. So that really conferred stability to the protein of interest. And by doing so, they could increase the load on that isolation step in the process. They could even remove the filtration steps, the ultra and diafiltration steps at the end of the process. And that saved a lot of hundred thousands of US dollars than when they were scaling up the process, as well as enabled them to utilize the protein A resin to a much fuller extent before repacking the columns. So this is just to show you one way of using DSC to complement the concentration measurements in the process with the SBR. And I think combining these two together really improves not only the quality of your product in the end, but also the productivity. But that's not all to it. Of course, when you're working with screening, you're generating a lot of data with these high throughput systems. And if you're doing further evaluation and data analysis, you're generating massive loads of data, just too much to shake a stick at, really. You need to manage that data in a convenient way and in a proper manner to keep control and track of all the results. And when you're coming to a point where you need to integrate the data, the situation gets even more complex. In order to do that, we have implemented in our systems a database, Oracle-based system to manage and sort the data and to enable import and export of data in a seamless manner to laboratory information management systems, all done under 21 CFR Part 11 compliance, which is of additional interest because these type of requirements are also getting highlighted by authorities. Not only the US FDA are looking at data integrity issues right now. This regulation is still in effect, so I think it's very important for those of you who are working in the GLP GMP space to look at those concerns, not only on the local installation, but also for the life of the analytical data outside the local installation and when you're integrating data together for decision making. With that, I would like to thank you so much for your attention, and if we have time, I'll be happy to take any questions now or later. Thank you.